Everyone, and welcome. Today, we are diving deep into one of the most incredible intelligence operations happening inside your body at this very moment. It's called Antigen Presentation, and it's all about how your immune system identifies a threat and activates the exact right soldier for the job.
All right, so here's how we're going to break this down. First, we'll look at the massive search problem the immune system has to solve. Then, we'll meet the expert cells that handle this mission. After that, we'll explore their two totally different strategies for reporting threats, depending on whether the danger is inside or outside our cells. And finally, we'll cover the critical safety check that makes sure we activate the perfect response every single time.
So first things first, let's talk about the search problem. You've got this huge army of T cells, right? But for any one specific bad guy, say a particular strain of the flu virus, the one T cell that can actually recognize and fight it is, well, it's unbelievably rare. It is truly a needle in a haystack situation.
And that really is the million-dollar question. I mean, a microbe can get in anywhere, your skin, your lungs. So how does that one specific T cell out of millions manage to find its enemy before it's too late? Just having T cells wander around the entire body hoping to bump into the right invader? That would be way too slow and totally inefficient.
And just to put some numbers on this, the chance of any single untrained T cell recognizing a specific antigen can be as low as 1 in 100,000 or even 1 in a million. The odds are just staggering. So you can see why the body had to come up with a much smarter strategy. And here is that elegant solution. The immune system doesn't send its entire army of T cells out on patrol. No, that would be crazy. Instead, it has specialized scouts capture the enemy, the antigens, and bring them to central command posts, which are your lymph nodes and your spleen. All the naive T cells are hanging out in these locations, so it's much easier and faster to find the one soldier who can recognize the threat.
So who are these incredible scouts? Well, they're called antigen presenting cells, or APCs for short, and you can think of them as the immune system's elite intelligence agents. And the most important, the absolute master of this whole operation, is the dendritic cell.
Dendritic cells are just perfectly built for this job. They're stationed right at the front lines, in your skin, your gut, your lungs, basically anywhere an invader is likely to show up. They've got these special receptors designed to just grab onto microbes. And once they capture an antigen, they literally pack up, travel to the nearest lymph node, and present their findings to activate those naive T cells.
And check this out. Here's a perfect example. These are Langerhans cells, which are a special kind of dendritic cell, and they're embedded right in your skin. They form this incredible vigilant network of sentinels at one of our body's biggest borders, just waiting to sound the alarm.
Now this is where it gets really clever. These APCs use two fundamentally different strategies to report a threat, and it all depends on where that threat is. Is it an inside job or an external invader? This table gives us a quick overview of these two pathways, which we call MHC Class I and MHC Class II.
Okay, let's break down that first strategy, the MHC Class I pathway. This whole system is designed to deal with threats that are already inside our own cells. You know, things like viruses that hijack our cellular machinery to make more copies of themselves.
So the goal here is absolutely vital. It's to take any foreign proteins that are being made inside the cell, chop them up into little pieces, and then display those fragments on the outside of the cell. It's basically like the cell raising a big red flag that screams, "Hey, something is wrong inside me, help!"
Okay, let's walk through it. First, a viral protein floating around inside the cell gets shredded into little pieces by a sort of cellular recycling machine called the proteasome. Next, those little fragments get pumped into the cell's protein factory, the endoplasmic reticulum. Inside, they're loaded onto these special display molecules called MHC Class I. And finally, that whole complex gets sent up to the cell surface to be inspected by the immune system.
Okay, so that's how we handle threats that are already inside. But what about all the bad guys lurking outside our cells, like most types of bacteria? Well, for that, we need a totally different strategy, the MHC Class II pathway. And the goal here is the total opposite of Class I. It's not about what's being made inside the cell, it's all about what the cell has captured and eaten from the outside world. The APC essentially gobbles up a microbe and then reports back on what it found. And the process is completely different. The APC swallows the microbe into this little contained bubble called an endosome. Inside that bubble, digestive enzymes chop the microbe's proteins up into fragments. Meanwhile, the cell makes new MHC Class II display molecules and sends them in another bubble to meet up.
The microbial fragments get loaded onto the MHC molecules, and that whole complex is what gets presented on the cell surface for a different kind of T cell to come and inspect.
Okay, so this next part is absolutely crucial. Just showing a T cell a piece of an antigen isn't enough to launch a full-blown immune attack. And that's a good thing. Think about it, our bodies are full of our own proteins, and we definitely don't want our T cells attacking us. So the system has this brilliant safety check built right in.
And this slide shows its safety check perfectly. For a T cell to get fully activated, it needs two signals. Signal 1 is the antigen itself, presented on the MHC molecule. That's the what. But to make sure it's not a false alarm, it also needs signal 2. This is a separate confirmation signal from the APC that basically says, yes, this is a real threat, you have permission to engage.
We call this second signal co-stimulation. And here is the truly brilliant part. The molecules that provide this signal only show up on the APC after it's been stimulated by the microbe itself, by recognizing things like bacterial cell walls. This is what ensures our T cells only get activated in response to an actual infection, not some harmless dust particle or piece of food. This two-signal system is a fundamental safeguard against our immune system attacking our own healthy tissues.
All right, let's get to the payoff. Why go through all this trouble? Why have two separate, complex presentation pathways? Well, it's because we have two major types of T cell soldiers, each with a very different job, and these pathways make sure we always activate the right one for the mission.
And it's just a perfect division of labor. The Class 1 pathway, which signals an inside job, activates our CD8+ killer T cells. Their one and only mission is to hunt down and destroy our own infected cells, essentially taking out the virus factories. Then you have the Class 2 pathway, signaling an external threat. This activates CD4+ helper T cells. They're like the generals of the battlefield. They don't do the killing themselves, but they orchestrate the entire response, helping other cells kill bacteria and telling B cells to make antibodies.
This slide just sums up the whole beautiful system. Up top, you see an internal threat from an infected cell presented on MHC class 1, leading to a killer T cell destroying that target. And on the bottom, an external microbe gets eaten, presented on MHC class 2, and a helper T cell is activated to orchestrate the defense. It is a perfectly tailored response, matched exactly to the kind of threat the body is facing. So, we've just seen this incredibly precise and elegant system for telling friend from foe, and for distinguishing between internal and external threats. But this level of precision raises a really critical question. What happens when the system gets it wrong? What happens when it accidentally targets our own self-proteins? Well, that, of course, is the basis of autoimmune disease, where this beautiful protective system gets turned against the very body it's supposed to defend.